Good morning, everyone. Can you all hear me, first of all?
Yes.
Okay, so I'm Sidra, and today I'm going to go through with you guys the chapter Carbohydrate Metabolism. So before I start, I want all of you to answer some questions, yeah? So that I know which topics I should focus on and which topics you guys know about, okay? Sorry guys, just a second. Please take out your phones and join the quiz. Is everyone in? Yeah, okay? So let's begin. You're not joining? Okay, let's begin. The first question is up here. Please answer so that we can discuss.
It says the voting is closed.
It says the voting is closed?
Maybe you press next twice. Yeah.
Okay. And done. The answer is 2. Okay? Not 3 because 3 in total, but how many molecules? They're just 2, which is glycerate 1,2 bisphosphate and fructose 1,6 bisphosphate. Sure? Okay. And the answer is right. I see most of you know the answer. Next question. Okay. Why is conversion of glucose to glucose 6-phosphate not committed? It's because both glucose and glucose 6-phosphate can go into other pathways. For example, the pentose phosphate pathway. What is the starting material? It's glucose 6-phosphate. Okay? Okay, I see that you all are good with this. Let's go to the next question. Phosphoglucose. Okay, it's the second answer. These are the type of questions, they're really direct. So once you go through, I think most of you have not gone through revise the chapter. Have you guys? Yes or no? So once you go through, you'll be good, okay? I agree. They're a little. It's okay. After the session, you're gonna be fine. Good. Good. Okay, I think this is the last question. Yes. Oh, we have more. Okay. Okay, why 4 fates? First is it can go into glycolysis, the pentose phosphate pathway, gluconeogenesis, and glycogenesis. Okay, 4 pathways. That's why 4 fates. Done, everyone? 2. Nice. Okay. Okay, it's a debranching enzyme. Okay, we'll go through this, so don't feel tensed or anything. We'll go through this. (50-50) versus glycerin 2-phosphate. Okay? I'm gonna explain all this, so it's fine. And this is just for an idea on what I should focus on. Yes. Not xylulose, it transfers 3 carbon units. Okay, the answer is 1. And the explanation for this is, read the question. It's from fructose 6-phosphate ending to glycerate 2-phosphate. Now, how many ATPs do we use in this pathway? From fructose to fructose 1,6-phosphate? 1 ATP. And then, moving forward, we have that reaction, glycerate 1,3-bisphosphate to glycerate 2-phosphate, where we are producing 2 ATPs. Yeah, because there are 2 glyceraldehyde molecules. Now, 2 ATPs produced, 1 ATP used. Net would be 2 minus 1, that's 1 ATP. Yes. It's isomerase because aldose to ketose conversion is an isomerization reaction. So from the reaction type, the name of the enzyme, which is isomerase. Yes. It's glycolysis, breakdown of glucose to form pyruvate, 2 pyruvates. The leaderboards, Suad, wow. And then comes Zain and Yasmin, and Malik, then Aisha. Good guys, really great, okay? Now we'll start with basic stuff. What's metabolism? It's basically the reactions within the cells, the set of reaction within the cells leading to a synthesis or a breakdown of a molecule. So why do we synthesize a molecule? We synthesize a molecule to use it for something, okay? And why do we break a molecule? It's primarily for energy production in the body, yeah. And it can be used in different ways. Now the main metabolic pathways are catabolism and anabolism. catabolism, breaking down, anabolism, building up. If you want examples, catabolism, an example for it is glycolysis, and there was a question right now. And then anabolism, example is gluconeogenesis, where you're building up from pyruvate to glucose. Okay? Now these are the pathways we are gonna go through today. Okay, so this is a simple summary about what happens to glucose once it enters the cell. Glucose enters the cell. It can either go and be stored as glycogen, or it can get into the pentose phosphate pathway where it's used for many things. Then it can go into glycolysis and be converted to pyruvate. And pyruvate has two fates, okay? One, in the absence of oxygen, it's turned into lactate, and I'm going to be talking about this. And in the presence of oxygen, it's going to be oxidized more in the citric acid cycle, which is, I think, the next chapter after this, yeah? Fine. And then glycogen, how do we obtain glucose in the body? By glycogen breakdown, that's glycogenolysis, or by gluconeogenesis. Okay, so let's begin. I'm going to go reaction-wise, mechanism-wise, so that we are in an orderly fashion. Fine? Do you guys understand until now? Yes. Sure, okay. If you guys don't get anything, tell me. If you guys want me to repeat something, tell me, okay? Now, first reaction in glycolysis is the synthesis of glucose 6-phosphate. Why does one synthesize glucose 6-phosphate? It's because glucose can diffuse through the membrane, okay? So to trap the glucose inside our cells, we add a phosphate so that glucose doesn't go out, yeah? So it stays inside the cell and we can continue with glycolysis. The enzyme, hexokinase, and this uses ATP, and yeah, that's it. And this reaction is irreversible, and that's it. Short and sweet. Now we go for the second reaction. That's conversion of glucose 6-phosphate to fructose 6-phosphate. Glucose and fructose are isomers. So this reaction is an isomerization reaction. And it's a reversible reaction. And the enzyme responsible for catalyzing this reaction is phosphoglucoisomerase. Isomerization reaction, that's why the enzyme responsible is also an isomerase. Okay? And there's an intermediate form in this reaction, which is an enediol intermediate. I will show you guys what's an enediol intermediate. It's basically two C's with a double bond, and both of the C's have an OH. This is an enediol. Okay? As you can see here, this is the intermediate in this reaction. Fine? Good? Can I continue? Okay. The third reaction is phosphorylation of fructose 6-phosphate. Okay? To make fructose 1,6-bisphosphate. This reaction is the most regulated reaction in glycolysis. Okay? And it's irreversible, and it's the first committed step in glycolysis. So the products of the previous reactions can go into other pathways, but this stays. It's lower limit. Okay? And ATP is required. So till now, how many ATPs have we used? 2. Right. That's it. And I think there is a mechanism, or there's not. No, there's no mechanism. I'm going according to the textbook, by the way. Yeah?
There's like 2 versions of the book. Which one are you following? Because I think there's some differences between each.
I'm not sure which version. But then I took it from your ear. I have no idea.
It's fine.
So I'll tell you, okay? I'll check and I'll tell you. If there is anything extra in the other version, I will send a video, okay? I'll send a video explaining that part for you guys. Now the fourth reaction is the cleavage of fructose 1,6-bisphosphate. Now some of you will ask me, why don't you just make glucose 1,6-bisphosphate and then cleave it? It's because it's difficult to add phosphate to the 6th carbon of glucose. That's why you change it to fructose 1-phosphate and then you phosphorylate fructose. The enzyme, okay, the type of reaction with this is an aldol cleavage. Cleavage, breaking of a bond. Wait, where am I? Okay, cleavage, breaking of a bond, and enzyme used is aldolase. Aldol cleavage and enzyme aldolase, same as the reaction. And this reaction is irreversible. What we do is we break fructose 1,6 bisphosphate to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. The mechanism, okay? So basically, this is from the textbook. The open change form of fructose 1,6 bisphosphate, that is this, okay, binds to the enzyme, the active site of an enzyme, by Schiff base formation. What is Schiff base formation? It's basically the Schiff base is this, C double bond N. Okay? This carbon over here, C double bond O, binds to the active site of the enzyme by Schiff's base formation. And then what happens is a general base within the active site removes a proton. What's a proton? H plus. So removes a proton at C4. So 1, 2, 3, 4. A proton, this H is removed. Okay, once this H is removed, the bond between carbon number 3 and carbon number 4 breaks. Okay? Once this bond breaks, glyceraldehyde 3-phosphate is cleaved. And the second step is basically cleavage of this Schiff's base formation. Like we formed this bond, yeah? Now we need to cleave it so that we get dihydroxyacetone phosphate. The cleavage of fructose 1,6 bisphosphate is thermodynamically unfavorable. That is, it has a positive Gibbs energy constant. I'll tell you what's Gibbs energy and I'll give you a trick, okay? So there are, Gibbs energy is like the free energy released by a reaction, okay? We have, if it's positive, we have if it's 0, and we have if it's negative, okay? If it's positive, the reaction would go reversible, okay? It would favor to go backwards. If it's 0, that means the reaction is in equilibrium. So the forward reaction is equal to the backward reaction. And if it's negative, that means the reaction is favored to go forward, okay? So if the delta G over here is positive, so how would this reaction proceed? Like, want to go, where would it want to go?
Backwards.
Backwards. So now you ask me, how is it going forwards? Because the products formed here are removed by the next reaction rapidly, so that it's forced to go forwards, okay? Now reaction number 5 is the conversion of glyceraldehyde 3-phosphate to dihydroxyacetone phosphate. Why is it done? Because glyceraldehyde 3-phosphate is the only molecule that can continue in glycolysis. Now, we don't want to lose 3 carbons in form of DHAP, so we change it, okay? We change it to glyceraldehyde 3-phosphate. And the enzyme responsible is triose 3-phosphate isomerase. Again, isomerization reaction, so the enzyme is isomerase, okay? Okay, this is the mechanism for this reaction. So basically, the glue 165 residue in the enzyme acts as a nucleophile and attacks this proton over here, and it takes it away, okay? At the same time, this residue, His 95 residue of the enzyme, donates its H here, okay? So this takes it away, and this donates. Takes it away at the first carbon, donates at the second carbon, okay? At the second, to the O of the second carbon, okay? Not at the carbon itself. So what is formed again? Enediol. OH, OH, okay? And then, His 95 residue, it wants its hydroxyl back, not hydroxyl, proton back. It wants its H back. But our dihydroxyacetone phosphate cannot give this H back. So what it does, it gives this H back. Okay, this H is given to His 95, and the double bond over here extracts or like snatches this proton from glue 165. Do you get it? Do you get it? Okay, I'll explain again. Okay. Basically, what are nucleophiles?
Negative.
Sorry?
Negative.
They're negative charge. So what do they love?
Protons.
Positive charge, protons. And electrophiles are positive charge. What do they love? Negative charge. Okay. Now, this acts as a nucleophile. That means it's negative. It loves protons, so it will snatch one from here, from the first carbon. This over here is an electrophile, so it wants to make itself negative. Okay, so it will give one of its positive charge to dihydroxyacetone phosphate at carbon number 2. And then enediol is an intermediate. Okay, and then what happens is this now wants its charge back. He doesn't like being negative, so he just takes a proton from the hydroxyl of the first carbon, and this likes being such way, but then our substrate wants the proton back. Okay, so it takes this proton away from glutamine 165 of the enzyme, forming glyceraldehyde 3-phosphate, which is the product. Okay, get it? Sure? Sure? Okay. So with this, we're done with the. Okay. With this, we're done with stage 1 of glycolysis, and this is the energy investment stage. As you can see, we did not get energy yet. Instead, we've been using energy. So how many energies did we use? 1, 2 ATPs. Sure. So we're done. And then there are 2 non-reversible reactions here. Other all are reversible. And we're done. Now reaction number 6. This is the oxidation of glyceraldehyde 3-phosphate. Okay? And oxidation and phosphorylation. So we oxidize it and then we add a phosphate. The product is glycerate 1,3-bisphosphate, which has a high energy phosphoanhydrate bond. This is the high energy phosphoanhydrate bond. This one over here. Okay? NAD plus is used and it's reduced to NADH. And this is a reversible reaction. Now this glycerate 1,3-bisphosphate has high phosphate transfer potential. Okay? So it loves to give away its phosphate, which we will see in the next reaction. Oh, before that, the mechanism again. Okay. I'd like to talk about the enzyme to begin with. The enzyme again is glyceraldehyde 3-phosphate dehydrogenase. It has 4 subunits, and each subunit has 1 binding site for the substrate, that's glyceraldehyde 3-phosphate, and 1 binding site for NAD+. Okay? Now we'll start with the mechanism. So what happens? Glyceraldehyde 3-phosphate enters, and it forms a thioester bond with the enzyme active site. Okay? So the thioester bond is here. Okay? It's like this. SC double bond O R. This is a thioester bond. Okay? Now this is how it's happening. It enters, and from here till here, what it's doing is basically forming a bond with the enzyme's active site. That's it, nothing else. But this is like S is attacking the C, and the electrons here are going to O. Okay? And then here, what's happening is resonance stabilization of this benzene molecule over here. And then a hydride ion, that is H negative, is released, which is taken up by the NAD plus present in the active site of the enzyme. So H minus, after thioester bond formation, H minus hydride ion is released, which is taken up by NAD plus. Okay? And this NAD plus goes out into the cytoplasm or wherever, and it's replaced by another NAD+. Okay? Now, if you remember the reaction, we also had a phosphate added in over here. Yeah? So what's the use of this phosphate? Is basically over here. The phosphate comes and attacks the thioester bond to cleave the bond between the enzyme and the product. And once the bond is cleaved, we get our product, which is glycerate 1,3-bisphosphate. Do you guys want me to repeat?
Yes.
Yes? Okay. So glyceraldehyde 3-phosphate forms a thioester bond with the enzyme. It's a covalent bond. You know what's a covalent bond?
Yes.
Sharing electrons, yeah? Now, where is this happening? All over here, till here. The sulfur group of the enzyme here attacks the carbon over here of the substrate, and then there's like a lot of resonance stabilization happening to form the covalent bond. Basically, okay, it attacked, now the bond is forming over here, and everything is stabilizing within the environment. And then here, after stabilization, the H- ion is released. It's extra, so it's released. And this H- ion is taken up by NAD+ over here, which is like, it's just present in the active site. Okay? And this NADH that is formed leaves, and another NAD+ comes in back to the enzyme's active site. And then what happens? A phosphate. Now, we did so much to make this bond, and now we have to break this bond so that the product can be released and it can go into glycolysis. So a phosphate comes and attacks the carbon here and forms a bond with the carbon, and this bond is broken and the product is released. What's the product? Glycerate 1,3-bisphosphate. Got it? Sure? 100%? So if you didn't get it, you can come to me after the session. Sure. Okay, so next reaction is a substrate level phosphorylation. What it is basically the product of the previous reaction, which is glycerate 1,3-bisphosphate. It loves to give its phosphorus away. Okay, it wants to give it away. So it has a high energy transfer potential. I've not written here, but high energy transfer potential. Okay, so it loves to give its phosphorus away to ADP, which loves to take phosphorus. It doesn't want to give phosphorus. So it's called low, it has a low energy transfer potential. So it doesn't want to give it away, it wants to take it. You understood? And ATP is synthesized in this reaction, and it's called a substrate level phosphorylation reaction. How many ATP per glucose? 2. Per glyceraldehyde, glycerate 1,3-bisphosphate? 1. Okay? And the enzyme, phosphoglycerate kinase. Okay? Anything with ATP is always a kinase. Now, okay, basically ADP, it acts as a nucleophile and it wants to take phosphorus. So it will attack and it will take away phosphorus from the phosphoanhydride bond that I mentioned before. And the product is glycerate 3-phosphate. And this, everything I mentioned, and this is a spontaneous reaction. Why? Because delta G is less than 0. So when did I say if it's less than 0, it likes to go forward. Sure. Okay. The next reaction is interconversion of glycerate 3-phosphate to glycerate 2-phosphate. Now glycerate 3-phosphate, it has one phosphate left, and it gets really like protective about it. It doesn't want to give it away. So we need to convert it to something that will want to give its phosphate away so that we can get energy. Okay? And that's why the next two reactions are happening, so that we get phosphoenol pyruvate. Now this reaction is the conversion of glycerate 3-phosphate to glycerate 2-phosphate, and the enzyme is phosphoglycerate butase that does this reaction, catalyzes this reaction. It's a two-step addition elimination cycle. So it is, okay, it has two steps and there is an addition and elimination. In the first step, phosphate, inorganic phosphate, is added to carbon number 2. This is added here. And there is an intermediate which is glycerate 2,3-bisphosphate. And then there's hydrolysis, that is water is added so that this phosphate is removed. Okay, so as you can see here, the phosphate here is removed and we get glycerate 2-phosphate. Okay? Now the reaction 9 is dehydration. Dehydration means removal of water. Okay, removal of water. Catalyzed by the enzyme enolase, and the product is phosphoenol pyruvate. Enolase. Okay? How does it happen? So this enzyme removes a proton, that is H, from carbon number 2. Carbon number 2, 1, 2. H is removed. Okay? And at the same time, the carboxylic acid side chain, which is present in the enzyme, adds H to OH over here. Okay? So H is removed from here and H is added here. So if H is added here, there's H2O here. Okay? And this is a good leaving group. Okay? It just leaves the reaction. So H2O will leave and we'll have phosphoenol pyruvate. Okay? So do you guys know about enol keto tautomerism? Okay, because you will need this for the next reaction and you can't. I'll use that word. You guys can see all that, yeah? So basically ketones, they like to be present in the atmosphere in two forms, the enol form and the keto form. Okay? This is the enol form. C double bond C, H, 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 OH. Okay? This is not enediol, there are no two OHs, but one OH, and it's the enol form. Okay? And this is the keto form. Okay, this is the keto form. Now this form over here is more stable. If it's more stable, the molecule would like to be in a more stable form rather the unstable form. Okay? So this is tautomerization. These are called tautomers because they have the same molecular formula and they keep changing, so it's tautomerization. Okay? I'll go into the next. As you can see here, okay, this is an enol form because C double bond C here. So it's an enol form, so it wouldn't want to stay like this. It will want to go into a more stable form, and that's why the next reaction happens, so that it can go into the stable form. Why can't it go to the stable form? Is because there is a phosphate group here, so it cannot form C double bond O. Okay, there's a phosphate group here, so now it will want to give away the phosphate. So if it wants to give away the phosphate, what's it called? High energy transfer potential. And who will it give it away to? ADP, which has a low energy transfer potential. The last reaction, that's synthesis of pyruvate by the enzyme pyruvate kinase. And basically ADP will attack this phosphorus of phosphoenol pyruvate, and it will readily give away. And once it gives away phosphorus, this is in its enol form. And then it is spontaneously it will switch into its keto form. That is more stable. And how many ATPs are formed for each molecule of glucose in this reaction? 2. That's it. You want me to repeat? Okay, done. So with this, we are done with the stage 2. So here, it's like a profit stage. So we invested our energy, now we've got our profit, that is 4 ATP, but like the net ATP would be 2 because we've invested 2 ATPs. And another energy source, that is NADH. Okay, 2 NADH. Okay, this is basically a summary of glycolysis. I'll be sending you these slides, so if you want to refer this, you can. It's a catabolic breakdown, anaerobic. It happens in the absence of oxygen. Glucose, which has 6 carbons, is broken down to pyruvate, which has 3 carbons. And the energy that is captured in ATP and NADH. 2 net ATP, 2 net NADH. And there are 10 reactions in total, and there are 2 stages: energy investment and energy production. Okay? And this is like an equation where it's summed up. Fine? Okay, so the pyruvate produced has 2 fates. Okay? Under the presence of oxygen, it will be converted to acetyl-CoA, which enters the citric acid cycle and electron transport chain to get oxidized and produce more ATP. In the absence of oxygen, it is reduced to lactate, so that NAD plus can be regenerated. Now the process in which NAD plus is regenerated is called fermentation. Okay, fermentation. And there are 2 types of fermentation. Okay, we have homolactic and alcoholic. Homolactic, lactic acid; alcoholic, ethanol. Homolactic fermentation is usually in the RBCs, lactobacillus bacteria, and mussels. What happens? Pyruvate and NADH plus will be, pyruvate is converted to lactate, and NADH is converted to NAD plus, catalyzed by lactate dehydrogenase. Okay, and this NAD plus can be used in glycolysis. And this other one happens in yeast and bacteria. What happens is pyruvate is converted to acetaldehyde, and while this conversion, they lose 1 carbon in the form of carbon dioxide. And then alcohol dehydrogenase converts acetaldehyde to ethanol, and NAD plus is reproduced. Okay. The regulation of glycolysis. It's controlled by allosteric enzymes. What are allosteric enzymes? They are enzymes that contain an active site for the substrate and an allosteric site for regulation, activation, or inhibition of that enzyme. Okay? The enzymes which are the allosteric enzyme from glycolysis are hexokinase 1 to 3, which are formed. There are different forms of hexokinase seen in all the cells. And there's another form, which is hexokinase 4, also called glucokinase, which is found in the liver only. Okay? And then we have PFK1, it's the most regulated. And then we have pyruvate kinase. Okay? The hormones that regulate glycolysis include glucagon and insulin. Okay? Now I'll explain these. Okay, this is the regulation of PFK1. Okay? PFK1 is activated by fructose 2,6-bisphosphate. Okay? This activates PFK1 in glycolysis. Okay? Now, how do we get fructose 2,6-bisphosphate? The answer is here. Okay? This enzyme over here, PFK2, converts fructose 6-phosphate to fructose 2,6-bisphosphate. Okay, and this goes and activates fructose PFK1. Okay, how does this happen? And this enzyme over here is regulated by the hormones I mentioned: glucagon, insulin. Okay, so how does it happen? Glucagon. Just a second. Sorry, guys. Okay. Glucagon binds to its receptor in the cell, initiates cyclic AMP, and then releases this enzyme called protein kinase A. Protein kinase A adds phosphorus to PFK. Okay? Phosphate to PFK, and this PFK is turned from active to inactive. So if it's inactive, it cannot produce this. And if this is not produced, PFK1 is inhibited. Okay? Cannot do it separately. At the same time, or maybe like different times, insulin over here enters the receptors of the cell, and then cyclic AMP and everything, and releases phosphoprotein phosphorylase. What does it do? It removes this phosphate group and makes PFK2 active again. So it can produce fructose 2,6-bisphosphate, which will activate PFK1. Do you get it? Sure. Okay. Now this is again these are from the slides, okay? So hexokinase, nothing activates hexokinase. Glucose or any other sugar enters the cell, it will phosphorylate, okay? But it's inhibited by its own product and ATP. ATP is an indicator of high energy. And then we have PFK1 activated by fructose 2,6-bisphosphate and AMP. AMP is an indicator of low energy. And it's inhibited by citrate and ATP. Both of them indicate high energy. Again, pyruvate kinase is activated by fructose 1,6-bisphosphate. Now, we know fructose 1,6-bisphosphate, okay? It's a product of the PFK1 reaction. What it does is, once it's produced, it activates pyruvate kinase, okay? So it's like a feed-forward mechanism of activation. 2,6-bisphosphate activates PFK1. PFK1 produces fructose 1,6-bisphosphate, and this goes and activates pyruvate kinase, okay? Feed-forward mechanism. Inhibited by acetyl-CoA and ATP, both are indicators of high energy. And that's it. Okay, I've mentioned everything. Do you want me to go through it again? Gluconeogenesis, it's the reverse of glycolysis, except the 3 irreversible reactions. You have like a bypass reaction for those 3 irreversible reactions, which I'm gonna talk about. First reaction, now we're going from down to up, okay, in the opposite way. So the first bypass reaction is synthesis of phosphoenol pyruvate. It requires 2 enzymes, so therefore 2 steps, okay, 1 and 2. And first reaction is catalyzed by pyruvate carboxylase, so it will use CO2, okay, and it's found only in the mitochondria, It converts pyruvate to oxaloacetate by adding CO2, and it uses 1 ATP, converts to ADP. So if you want to get 1 glucose molecule, this reaction will happen twice. So 2 ATPs will be used. And the transfer of CO2 requires the coenzyme biotin, which is bound to the enzyme's active site. Okay? The mechanism is pretty fine. Now the keto form of pyruvate is keto form, yeah? More stable, so it will stay in keto form. It's turned into enol form, okay? And then, okay, I'll show it again. The keto form over here is turned into this form. And while it's going back to this, the double bond attacks CO2, okay? And this forms a bond forming oxaloacetate. Yes. Okay, yeah. So as we know, biotin is bound to the CO2, okay? So what happens? Electrons from this double bond attack CO2. Biotin releases this carbon dioxide, which goes and attaches. Sure? Fine? And then we get oxaloacetate. Now the second reaction is basically conversion of oxaloacetate to phosphoenol pyruvate. Again, it uses another form of energy, that's GTP. So until now, 1, 2, 3, 4 energy molecules used. And it's decarboxylated, removal of carbon dioxide, and phosphorylated, that is adding of phosphate. Uh, it's driven by GTP hydrolysis, that's GTP to GDP. Okay, now this enzyme over here, in humans it's present in both mitochondria and cytoplasm, okay? But in some cells and in some animals, it's not present both the places, okay? It's not present both the places. So how does it like work? Like, because this oxaloacetate here cannot cross the mitochondrial membrane. So if we wanted to go to the cytoplasm, how will it go? It will go through the malate shuttle. Now I'm going to talk about malate shuttle in the next slide. Did you get this? Okay. Malate shuttle, oxaloacetate is turned into malate by malate dehydrogenase. NADH is used and converted to NAD+. Now what's the significance of malate? It can cross the membrane, so it will go outside, and then there is a malate dehydrogenase in the cytoplasm, which will catalyze the reverse reaction, and malate is converted to oxaloacetate, and NAD+ is converted to NADH. Okay? And this NADH can be used in gluconeogenesis. In which reaction? In this reaction over here. Glycerol 1,3 bisphosphate to glyceraldehyde 3-phosphate, where NADH is used and converted to NAD+. Okay? Okay? Sure? Okay. Now the next reaction is conversion of fructose 1,6 bisphosphate to fructose 6-phosphate, and it's an energy releasing reaction. But this energy is not released in the form of ATP. Okay, instead the inorganic phosphate is generated. The reaction is irreversible and is catalyzed by this enzyme, fructose 1,6 bisphosphate, and it's activated by citrate, that is high energy, inhibited by AMP, low energy. Low energy gluconeogenesis won't take place. Instead, glycolysis will take place. Okay, and then we get fructose 6-phosphate. Now the last bypass reaction in gluconeogenesis is formation of glucose from glucose 6-phosphate. What's the enzyme? Glucose 6-phosphatase. And it's only found in the liver and in the kidneys. Only in the liver and in the kidney. And the reaction is irreversible. And what it does, it basically hydrolysis, adding water to break the bond. Hydrolysis of glucose 6-phosphate to release glucose, and again inorganic phosphate and not ATP. Okay? And then this glucose is released into the blood. Now a summary, 6 ATPs are used, 4 ATP and 2 GTP. At the same time 2 NADHs are used. It's an anabolic process that is building up, and energy is consumed rather than, liberated. Do you guys want me to stop? Okay. Now there are 3 substrates for gluconeogenesis: lactate, glycerol, and alanine. Okay? Okay. Now this is found in RBCs and cells that lack mitochondria. Okay, and if they have like low oxygen concentration, so it's going to be pyruvate in glycolysis. It's converted into lactate. So this is found in cells like that. Now, how does it work? For example, let's take muscles. When we exercise, the oxygen concentration in muscles goes down, and lactate is produced by glycolysis, since muscles need energy for moving. Now, this lactate released by the muscles goes to the liver, where it's converted back to pyruvate and by gluconeogenesis to glucose. And yeah, this is the Cori cycle. And this glucose goes back to the muscles to generate energy. Next we have glycerol. Glycerol is a product of fat metabolism in the adipose tissue. Glycerol in the liver is converted to dihydroxyacetone phosphate, which can be used for glycolysis again. How? Which can be used for gluconeogenesis. Okay, gluconeogenesis. Glycerol by glycerol kinase to glycerol 3-phosphate, by dehydrogenase to dihydroxyacetone phosphate, and gluconeogenesis continues. Okay. Lastly, the most important substrate is alanine. Alanine muscles produce pyruvate, right, while exercising, and some of it is converted to alanine by this transaminase reaction, which you guys will learn in like later chapters. Okay, pyruvate and glutamate come together, and this reaction would catalyze your transaminase, yields alanine and alpha ketoglutarate. What happens is this alanine is gone to the liver from blood, and this alanine is turned into pyruvate to glucose, gluconeogenesis. So what's the purpose of this cycle? It's to restore the amino acid balance. It's to restore the amino acid balance in the liver. Okay? Now the regulation. It's usually with allosteric enzymes, hormones such as cortisol and insulin. And what are the allosteric enzymes? We have this one over here, pyruvate carboxylase. This one over here, fructose 1,6-bisphosphatase, and then that's it. Two allosteric enzymes. Hormones, cortisol and insulin. Okay? Insulin would inhibit or activate gluconeogenesis. Inhibit. Cortisol does the opposite. Okay, and availability of the substrate. So if the substrate is not available, it can go with gluconeogenesis. That's it. And then we are at our pentose phosphate pathway. This pathway does not generate ATP. So why is it used? It's just used to produce these products over here that are NADPH, ribose 5-phosphate, and these are required for anabolic processes and for the production or like a structural component of nucleic acids. Okay, NADPH for anabolic processes, and it's a powerful antioxidant. So cells that are prone or like at risk of oxidation, such as RBCs, they undergo pentose phosphate pathway so that they produce this, which is an antioxidant. And this is for like DNA synthesis. Okay, DNA and RNA synthesis. Now this pathway has two phases: oxidative, non-oxidative. Oxidative, two NADPHs are produced, and all the reactions in this phase are irreversible. Okay. Non-oxidative, it produces three intermediates: ribose 5-phosphate for nucleotide and nucleic acid synthesis, fructose 6-phosphate and glyceraldehyde 3-phosphate that can go into glycolysis. Okay. Now the oxidative phase has three reactions, and the third reaction is divided into two reactions, but it's the same enzyme. Okay. First is oxidation of glucose 6-phosphate, and it gives us NADPH and 6-phosphogluconolactone. What's this reaction over here? The second reaction is it's hydration. Hydration reaction, and the product we get is 6-phosphodegluconate, and the enzyme is gluconolactase. The last reaction is this, and it's a two-step reaction, but it's the same enzyme for both of this. So first product over here is 3-keto-6-phosphodegluconate, and the second product and the last product of the oxidative phase is ribulose 5-phosphate. Okay. Now the non-oxidative phase, we have main enzymes that are working over here is ribose phosphate isomerase, ribulose phosphate 3 epimerase, transaldolase and transketolase. What do transketolase and transaldolase do? Transketolase are a TPP requiring enzyme. TPP's coenzyme form is also called vitamin B1. Okay? So it's vitamin B1 requiring or TPP requiring. And what it does, ketolase, so it transfers 2 carbon units from ketose to aldose. Ketose sugar to aldose sugar. And at the same time, this one transfers 3 carbon units from ketose to aldose. Okay? And that's what happens in the next few reactions. Now for this phase to start, we need 3 molecules of ribulose 5-phosphate. How many? 3 molecules. Okay? And this ribose phosphate isomerase will produce 1 ribose 5-phosphate from this. And ribulose 5-phosphate epimerase will produce 2 xylulose 5-phosphate here. This reaction is an isomerization reaction. And this is like a change. These both are epimers. You know what are epimers? So the one of the chiral centers is switched. Okay? And then ribose and xylulose again react. Transketolase is the enzyme. So what would it do? Transfer 2 carbon units from ketose to aldose. So 2 carbons from here are transferred here, resulting in glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate. Okay? Again, both of these react, and the enzyme catalyzes is transaldolase. Transaldolase, what does it do? 3 carbon units from ketose to aldose. So 3 carbons from here to here. Okay? Forming, this is the reaction, sorry. So 3 carbons from here to here. Okay? 1, 2, 3, from here to here. So the leftovers would be erythrose 4-phosphate and fructose 6-phosphate. And if you remember, I said there are 2 xylulose molecules produced. Okay? So where does the second one go? Okay, it reacts with erythrose 4-phosphate, transketolase again, so two of these are transferred to these, making fructose 6-phosphate and glyceraldehyde 3-phosphate. Okay? And that's it about this. There was no mechanism mentioned in the textbooks. Did you have any mechanism mentioned in your version? Okay. And then we have the fructose metabolism. Metabolism of other sugars, basically. And then we have, in other sugars, we have fructose, mannose, galactose, which we are going to talk about. So fructose metabolism usually happens in the liver. Sources of fructose are honey, fruits, etc. What happens is fructose is converted to fructose 1-phosphate by fructokinase, and this is then broken down into dihydroxyacetone phosphate and glyceraldehyde. Okay, because it just has 1 phosphate, so this phosphate goes to DHAP. And glyceraldehyde then is again phosphorylated to glyceraldehyde 3-phosphate, which enters glycolysis. Here, the 2 regulatory steps are bypassed. The one by hexokinase and the other one by PFK1. Okay? So it's like not that much regulated like glucose, but it is a little bit. Okay. Apart from that, mannose, hexokinase, mannose 6-phosphate, 2 fructose 6-phosphate, and then glycolysis. Okay? Here nothing is bypassed. But here 2 steps are bypassed. So this glycolysis with fructose has just 8 steps. But with other sugars it's 10. Okay, glucose and mannose and all that's 10. This I'll be explaining in the next, but let's read. Okay, let's do galactose. Galactose enters the cell, galactokinase to galactose 1-phosphate. Okay, this galactose 1-phosphate is turned into UDP galactose by uridyl transferase. This is changed into glucose by epimerase, UDP glucose by epimerase, and this UDP glucose is then converted to glucose 1-phosphate by UDP glucose phosphorylase, and this would either go into glycogenesis or glycolysis. Okay, which I'm going to talk about later. Next, glycogen metabolism. What is glycogen? It's the storage form of glucose. Basically, the synthesis of glycogen and degradation is carefully regulated, because we can't synthesize more glycogen than required. So if you do that, our other body cells won't get enough glucose. Yeah, and it's controlled mainly by 3 hormones: insulin, glucagon, and epinephrine. Okay. So there are 3 reactions. The first reaction occurs when glucose levels are high in blood, and the first reaction over here happens by the enzyme phosphoglucomutase, and it's a reversible reaction. Glucose 6-phosphate is converted to glucose 1-phosphate by this enzyme. There is a mechanism which is here. So the enzyme has a serine residue, okay, in which phosphoryl group, the P group, is attached. And this group is then transferred to glucose 6-phosphate to give us the intermediate, that is glucose 1,6-bisphosphate. Okay, I'll show you. So we have glucose 6-phosphate here, and the enzyme has a phosphate in its serine residue, and another phosphate is added from the enzyme at this point in glucose. Okay, giving us our intermediate glucose 1,6-bisphosphate. And as this bond is formed, this bond is broken down, and it releases glucose 1-phosphate.
I have a question. Whenever it's mutase, there's this type of intermediate where you add the phosphate and then you break it from the other side. When it's mutase enzyme,
Mostly,
because in glycolysis also it was like this. But isomerase is different.
isomerase is different. Yeah, you can say, because we're changing the position of something in mutase. We're changing the position of
The
phosphate here. Yeah. So there is this intermediate. Yeah, in the reactions we've gone through, all of them have an intermediate when it's mutase.
Oh, okay.
Yeah.
Yeah.
So I talked about the mechanism. And then the second reaction is synthesis of UDP glucose. Why does this happen? Is because UDP is a good leaving group. Okay, and then if you want to make glycogen, we need to form glycosidic bonds between two glucoses and more. Okay, so if we want to do that, we need more energy, but we can't use that much energy. So what we do is we add a good leaving group, that's UDP, to glucose, so that UDP leaves, two glucose form a bond, and we can form glycogen easily. Okay, so this is the reaction, and the enzyme is UDP glucose pyrophosphorylase, and it converts glucose 1-phosphate to UDP glucose, and this is called pyrophosphorylase, is released. Now this reaction has a tendency to go backwards. So what do? We hydrolyze this and we remove it so that this reaction goes further. We did that before, yeah? Positive delta J value, okay? And then the reaction number 3 is last reaction, which is synthesis of glycogen, and it has two enzymes that are participating, glycogen synthase and amylo alpha 1,4 to 1,6 glucosyl transferase, which is also known as the branching enzyme. There's one thing I need to say here is to synthesize glycogen, these enzymes need like a pre-existing glycogen molecule. They can't synthesize it from scratch. So what happens? Okay. Now we have the glucosyl residue. The glucosyl residue. Okay. It is linked to a primer protein. A primer protein called glycogen. Okay, it's linked to a primer protein called glycogen through the TYR194 residue. Okay, and this residue, and then what happens? More glucose, more UDP glucose are added to this glucosyl residue by the enzyme glucosyl transferase. So basically it's transferring all the glucoses there. So that's why it's called transferase. Okay? And once there are like 12 glucose bonded to each other by alpha 1,4 glycosidic linkage, these enzymes over here start their job. Okay? So what does glycogen synthase do? It basically keeps on adding UDP glucoses in alpha 1,4 bonds, okay, glycosidic bonds. And what does the branching enzyme do? Okay, I'll tell you what it does from the name. Okay, 1,4, it breaks the alpha 1,4 bond, and then 1,6, it forms an alpha 1,6 bond. Okay? So you can see here, it's breaking this bond, and then it's forming alpha 1 6. Okay? Do you guys get it? You want me to like repeat?
Can you repeat the part with the glycogenin and the glycosyl?
The glycogenin?
Yeah.
Okay. So basically, glycogenin is a primer protein. Okay. Now for our glycogen synthase and what's the other? The branching enzyme to work, we need like a chain of 14 glucose attached to each other. Okay. Now they can't work unless these 14 glucose are attached to each other. So who does this? Okay, who does this is basically this primer protein and this enzyme which is glucosyl transferase. Okay, so glucosyl residue, that's like UDP glucose, okay, goes and attaches to this residue in the enzyme. Enzyme has something called PYR194 residue, and this goes and attaches here, and it's accompanied by this primer protein. And then what happens is this enzyme over here adds more UDP glucoses, okay, forming like a chain of alpha 1,4 glycosidic bonded glucose, okay. And then after there are 14, our other enzyme work. You get it?
Yeah, so does that enzyme and glycogen synthase have the same function?
No.
No.
Similar function, but this works with the primer.
Oh, okay.
Glycogen synthase does not need the primer. Okay? That's it. And the branching enzyme is clear?
Yeah.
Clear? Okay. Next thing is breaking down of glycogen. So now we know how glycogen is produced. Now how is it broken down? It's by two enzymes again, glycogen phosphorylase and amylo alpha 1 6 glucosidase. Glycogen phosphorylase, what does it do? It uses phosphate and it breaks the alpha 1 4 linkages, so the linear linkages, and it releases this glucose 1 phosphate. Now what happens to this glucose 1 phosphate?
Glycolysis.
Yeah.
Phosphate powder.
Yeah, it can be changed to, yeah, exactly. And this stops when it comes to a branch point with 4 glucose residues. Okay, I will explain this branch point here. Okay, so basically, okay, so glycogen phosphorylase, it's working. Okay, linearly it will work properly, but when it comes to a branch, it will break all the alpha 1 4 branches bonds until it reaches this part where 4 glucosyl residues are attached to a branch. Here it will call its friend, the branching enzyme, amylo alpha 1 6 glucosidase. And what does this do? Okay, amylo alpha 1 6 glucosidase will take 3 of these residues all together and it will add them to the linear chain here. Okay, it will take this and add it to the linear chain, and then this one over here, it will break this bond by hydrolysis. Okay, it's not mentioned. Okay, it's not mentioned. Okay, it's just, okay, by how, it's not mentioned, but it breaks this bond and it releases this as free glucose. Okay, free glucose. Again, at a branch point, that is 4 glucosyl residues attached to glycogen at 1 6 by 1 6 linkage. The 3 of this attached by 1 4 linkages are cleaved and attached here to the linear chain, and this one left is broken off and sent as free glucose outside into the blood, and it can go into glycolysis. Regulation, glucagon activates glycogenolysis, because what's the aim of glucagon? It wants glucose in the blood, so it will break glycogen. Insulin inhibits because insulin wants glucose entering the cells. And then epinephrine activates glycogenolysis and inhibits glycogenesis. Okay? And by this we come to the last of our presentation today, which is the Crabtree effect. Now the yeast Saccharomyces cerevisiae, can ferment sugar to ethanol. Okay, nothing new. We know everyone ferments sugar to ethanol in large amounts. Why does it do it? To dominate its environment and competing with other microbes. Okay, so the fittest one survives. That one. What's Crabtree effect? It's even though oxygen is available, what does this yeast do? It ferments sugar and forms ethanol. So that's like the Crabtree effect. Now, when glucose and fructose levels are high, it will ferment the sugar and produce ethanol. And once the glucose levels are low, it will absorb all this ethanol, change it to acetyl-CoA, and converts it to acetaldehyde, and it's used for energy. Okay, and this makes it highly adaptable and able to survive in changing environments. Fittest one survives. Okay, and that's it. Thank you, guys.
Thank
If
you.
you have any questions, please let me know. Okay.